Hello. 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 Hello, Avinash. Hello. Hello. It is
Hello.
audible to you, Avinash.
Yes, sir.
Okay, we'll start
Yes, sir.
now. Okay, we'll start now.
Yes, sir.
Okay, okay. Hello. Aliva, it is audible to you?
Hello, yes, sir.
Okay, we'll start now.
Yes, sir, it's audible. Okay, sir.
Okay.
Yes, yes, sir.
Hello, Aliva. Screen is visible
Yes,
to
sir.
you? Screen
Yes,
is
sir, yes, sir.
visible? Okay,
Yes,
I
sir,
will.
visible.
Okay. Okay, good morning to all of you. So today we'll discuss about stereochemistry. Before going to start stereochemistry, we'll go for isomerism. What is isomerism? Then different types of isomerism. Then one by one we'll go the stereochemistry. So you have all idea about isomerisms. You have already studied in third semester maybe. So isomers which have same molecular formula but different in the arrangement of atoms. Okay, and the phenomenon is known as isomerism. So isomerism again may be categorized into two types. One is structural isomerism, another is stereoisomerism. So structural means there may be change in the structure, but molecular formula is same. But stereoisomerism means the molecular formula is same, but different in the arrangement of atoms in space or in three-dimensional space. So these are different basics or introduction to the isomerisms. So these are the classifications. So it may be categorized into structural and stereoisomerisms. Structural isomerism may be different types. One is chain isomerism, position isomerism, functionalism, metamerism is also there. It is called tautomerism. So under that you can get ketoenol tautomerism. Then you can go for stereoisomerism. It is two types mainly. One is geometrical isomerism, another is optical isomerism. So you can see the definition here. So structural isomers have same molecular formula, but different in the arrangement of atoms in a molecule. So first one is chain isomerism. So chain isomerism is they have same molecular formula, but differ in the arrangement in the chain. So here you can get the example like pentane. So pentane means it is a straight chain. So here you can see isopentane, here it is having branched. Then neopentane. So all are having same molecular formula, but different in the arrangement of atoms, or you can say groups in a molecule. Then you can go for positional isomerism. So here same molecular formula, but different in the position of the groups. So here you can see the example of propanol. So here OH group is attached at one position. So it is propanol 1, but here propan 2-ol. Here OH is attached on second position. So only position of the functional group will be changed. That is positional isomerism. Here another example is butanol, butane 1-ol or butane 2-ol. Only position of OH group is changed. So like this you can give some drugs also. Examples is enfluorine and isoflurane. This is the biological active drug. So here you can see the position of fluoro groups or chloro groups. These are changed. In enfluorine you see the position here. Here is different positions. The position of functional groups is changed. So that due to change in the position of groups, there may be change in the activity, or any biological property or chemical properties may be changed. Just I'll ask, it is audible to you? Oliva, it is comfortable?
Yes, sir.
It is audible to you all? Hello?
Sound lagging is there, sir. Hello,
What,
yes,
what?
sir. Sir is audible, but sometimes sound lacking. Hello.
Sound is yes. That's why confirmation is required. Okay.
Yes, sir.
This sound is coming late or what?
No, sir, like blur sounding.
Okay, maybe network is problem, maybe. Screen is
So now
visible
it's
to...
okay.
Okay. Screen is visible, na?
Yes, sir.
Okay.
Yes, sir. Screen is visible.
Okay, anytime, any doubt, you can raise the hands. Then I can understand any problems.
Okay, sir. Yes, sir. Okay.
Okay, this is another example of drugs which is having positional isomerism. Next, we'll go for functional isomerism. So functional means the functional group will be changed here. You can see the examples. So functional group will be changed. Here it is alcohol, but here it is again ether. So same molecular formula, but differ in the functional groups. That is called functional isomerism. Next, another example we can give, ketone and aldehydes. So propanone is one of the example, and its aldehyde is propanol. So this is the difference in the functional group, but same molecular formula. That is called functional isomerism. Next, we'll go for metamerism. So metamerism is same, molecular formula is same, but differ in the arrangement of alkyl groups on either side of this functional group. Or here we'll see example of methoxypropane and ethoxyethane. So here you see the alkyl group on both sides of the oxygen. Here it is unequal distribution of alkyl chain, but here it is equally distributed. So this type of isomerism is called metamerism. Next we'll go for tautomerism. So here it is simply the keto and enol form. Both are a reversible process with same molecular formula. That is called tautomerism. So these are all about your isomerism. I will discuss because it will reflect your designing the new molecules. So that is most important before going to stereochemistry. Next we'll go for stereoisomerism. Stereoisomerism is nothing but called spatial, not spatial or any pronunciation you can do, spatial or spatial isomerism, because the molecules in which the atoms are arranged in a three-dimensional space or 3D orientation of the atoms. That is called stereoisomerism. Suppose I have given the example of allopyrinol. So here you see this is 2D structure, and this is 3D structure. You see the difference. So 2D structure you can draw easily on your hand, but 3D is not possible by the hand. It can be possible by the computational process or by the help of softwares, or ChemDraw software is available. I'll show you the video how to draw these structures, then you can get the idea. Then here you see the different colors are visible. So here this color is representing carbon, here white color representing your hydrogen, blue color represents nitrogen, and this red color is oxygen. So by this way you can identify the different atoms present in a molecule arranged in three-dimensional space. So this is related to your stereoisomerism. So just you see the video, I'll show you. How the structure is drawing by the ChemDraw. This is 2D only. So you have to paste this structure into 3D space. How they are putting in 3D space. So this is visible on 3D screen. So this is your 2D. This is your 3D structure. So this represents your hydrogen, red color is oxygen, these are the carbons. So this is your salicylic acid structure. Okay, you can rotate in space also. You can save this one as a file with different names are there. You can save it. Thank you. We will go for next. Okay, now we'll go to next one by one for stereoisomerism. Yeah, next. So it is again classified, or we can categorize it into geometrical isomerism, which is again subdivided into cis and trans. And also another type of configuration is there. It is called E and Z system. So one by one we'll discuss. Then it is optical isomerism, enantiomers and diastereomers. So one by one we'll discuss. So first we'll go for geometrical isomerism. So geometrical isomerism is also called as configurational isomerism because the atoms are configured on the double bonded carbon atoms. Here you can see, suppose cis isomers, here similar groups like here hydrogen or methyl groups are present on the same side of double bonded carbon atom. This is written, identical side groups are present on same side of the double bonded carbon atoms. Then we can say it is a cis form. If similar groups are present across the double bond or opposite side of the double bond, then it is called as trans form. So based on this, we can design some drug molecules because in trans form, the bulk groups are away from each other and these are more stable as compared to cis, because in case of cis, there will be steric repulsion, so these are unstable. So most of the drugs are mainly trans form. So I'll give one by one examples are there. Next slides. So this is another example of malic acid and fumaric acid. So fumaric acid is in trans isomer and malic acid is cis isomer. In GPAT, most of the question is coming like this. So which of the following is a trans form, fumaric acid or malic acid? So you can give fumaric acid is the trans isomers. So in place of CH3 group, CaOH group is attached. So that is called fumaric acid. It is in trans form, which are present across the double bond, but COH group is present on the same side of the double bonded carbon atom. So here you see how the steric repulsion is more in case of cis isomers. So bulky groups are present on same side, or they are very near to each other or close to each other. That's why more repulsion will be there. So it is less stable. But when we compare with trans isomers, groups both are present away from each other. The distance is more. And here, this is not a bulky group. Only methyl is the bulky group. So there is less repulsion. So that this is more stable. Similarly, we can apply this one with examples of stilbestrol, that is diethyl stilbestrol. Diethyl stilbestrol. So ethyl group here you see in cis form, it is on same side, and this other groups are on opposite side. So here it is very more, or you can say more repulsion with each other. So that is, this is very inactive, or you can say it is 7% active, but here it is 100%. That is estrogenic property will be there. So this is transformed. So in this case, the bulky groups are away from each other, so that it is more estrogenic as compared to cis form. So like these examples or questions you can get in GPAT exam. So trans or cis form of dimethyl stilbestrol is, which form of diethyl stilbestrol is estrogenic, means transform is more estrogenic. So answer will be transform. Like this is another nomenclature, or you can say another deviation from cis and trans definition. We have studied cis means you have similar groups on same side of double bonded carbon atom, and in trans means similar groups are opposite side or across the double bonded carbon atom. But here some deviation from the rule. Here it is written, it is a square planar molecule in which NH3 groups or amino groups are attached on same side of the square planar. Or here in case of trans, it is in opposite side. NH3 or Cl. These are opposite side. So depending on that also, they are different biological activity. So in case of cis, it is chemotherapeutically active, but in case of trans, it is chemotherapeutically inert or inactive. So this is another example or another type of configuration in case of cis and trans. Hello, Avinash.
Sir.
It is audible to you all?
Yes, sir, audible.
Okay. Any problem, you can raise your hands here. Okay, on the screen.
Yes, sir.
Oliver, it is okay?
Yes, sir.
Okay. If anything, you raise your hands. Okay.
Okay. Yes, sir.
You people you can use your video also. You show your face. I am not able to find out here. Okay, next we'll go for EZ system. So it is another type of geometrical isomerism, but depends on the configuration of atom or group about the double bonded carbon atom. So here symbol Z is representing zusammen. It is a German word which means together, whereas symbol E represents opposite. It is the German word entagenen. That is the meaning of opposite. So here it depends on the priority of the groups. Suppose we'll consider here hydrogen, fluorine, bromine, chlorine. In periodic table you can see the molecular weight of hydrogen is less than fluorine, fluorine is less than chlorine, then bromine. So highest priority is bromine and chlorine. So here you see it is written already, fluorine has higher priority and bromine also higher priority. If higher priority groups are present on same side of the double bonded carbon atom, then it is called as like this, Z form. Z means together, means these are present on the same side. Here fluorine and hydrogen, they are considered as lower priority. So that's why they have lower priority and present on same side of the double bonded carbon atom. They are considered as Z form. Here you can see higher priority like bromine and chlorine, we have considered as higher priority based on the molecular weight. So they are present across the double bond or opposite side of the double bond. That's why it is called as E form, means they are present on opposite side. So like this E and Z configuration or geometrical isomerism you can consider like this. So like this you can take other examples also. Then we'll go for optical isomerism. So for a optical isomerism, the compound or a molecule should have ability to rotate the plane of polarized light. So how they can rotate the plane of polarized light? Means they should have chiral centers or asymmetric carbon. If there is no asymmetric carbon, they cannot rotate the plane of polarized light. So what is asymmetric carbon? Asymmetric carbon means they should have attached with 4 different groups. Here you can see the carbon which is asymmetric carbon. It is attached with 4 different groups. Then only it is called as optically active. Otherwise it cannot rotate the plane of polarized light. Then we will see what is plane of polarized light. Plane of polarized light means here you can see this is a general light source, or you can say ordinary light, which have wavelength in different directions. These are called as polychromatic radiations. They have different wavelengths. So when they will pass through a polarizer, so they will vibrate in single wavelength. So this is called monochromatic light. Then when it is passed again through a sample tube containing your chiral compounds, this is your sample tube which contain the chiral compounds. So they can rotate this light in a clockwise direction or anti-clockwise direction. If clockwise direction, then you can say plus or small d. If it is anti-clockwise direction, then minus or small l. So I have already discussed in class also in previous semester, small d small l and capital D capital L. That is different terms. You can see carefully. So here this is only small d and small l represented as plus or minus, depend on the rotation. So this is due to presence of chiral compounds only. But if it is not having chiral compound, that is achiral, so there is no rotation by the compounds because they have no capacity to rotate the plane of polarized light. So it is only a single wavelength, but there is no change in the rotation. So this is the limitation of achiral compounds. Only the chiral compounds they can rotate, and they should have some biological actions. That's why we are reading or we are going through the stereochemistry, because most of the compounds are having stereo centers or asymmetric centers. Due to the presence of chiral carbons or asymmetric centers, they are producing some specific biological activity. Why they are producing biological activity? by interacting with specific receptors. In this kind of carbon, they are able to bind with specific site of a specific receptor to produce the biological activity. That's why it is a very focused area in the pharmaceutical research. So carefully you can do the work on this specific area of research. So then we'll go one by one some examples here. You have studied in chemistry of natural products, carbon. So both are mirror image also, and they can rotate the plane of polarized light. So it is represented as plus form, plus carbon. Here it is minus carbon. So both are same structure, but they have different rotations depending on the presence of groups. So here it is plus form, this is minus form. It is smell like caraway, but it smell like a spearmint. So their activity will be changed depending on the rotations by the plane of polarized light. So here specific rotation is plus 62.5, here it is minus 62.5. So due to these properties, they have different activities also. So like this, some other examples also there. So this is another drugs which is having effective to prevent nausea and vomiting, but it is not a drug. Means this drug is having less activity, poor drug. So this both are mirror image with each other. So next we'll go for RS system. After EZ, RS is also another important configurational isomerism. So based on the scientist Kahn, Illock, Prelog, they have devised a system of nomenclature for the stereoisomers. One is called R system, another is S system. So how they will design this R and S? So depending on the priority of the atom or groups around the chiral center. This is your chiral center. So depending on the molecular weight, it is written that based on the atomic weight of the atoms. Okay. So here you see, so like this, anti-clockwise, it is increasing the molecular weight. So you can say it is a anti-clockwise or S form. But here you see, it is a clockwise direction. Then it is called as R form, from lowest to highest. From lowest to highest, that's why it is called R, clockwise direction. So this is another type of nomenclature for stereoisomerism. So you can see some examples here, like S-ketamine, R-ketamine. So both are same drug, but due to this R and S form, they have different activities. You see here it is anesthetic, but here it is a hallucinogen. So both are same drugs, but depending on this configuration, they are different activities. Just a second. Okay, so next we'll go for you see another example is also there, same R and S system, limonene. This is a natural product. So here it smells like orange, but it smells like a lemon. So here you see the properties, chemical properties is changing. And the specific rotation is plus 126.3 degree, here minus 126.3 degree. So depend on the priority of the groups, they have assigned as R or they may be assigned as S. Okay. So I have already told you how the chiral molecules are biologically active. Here you can see the figure. So this is a chiral center attached with 4 different groups. If it is attached with 4 different groups, they are completely binding with receptor site. This is the site. You can see here. So these are the receptor sites of the target proteins, or you can say any disease having some target sites. So to these sites, they are attached properly so that they are giving some response or biological activity. But here they are not properly. These are the achiral molecules. They have no proper arrangement of the groups. Here, different groups are not there, so that it is not bind properly with the receptor site. So there is no biological response here. So here you see the receptor site is different and your molecule shape is different, so that they are not able to bind properly. But here, depend on the receptor site, the molecule is fitting properly. Here you see. But in this case, it is not properly fitting with the receptor sites. That side is not giving your suitable activity, or there is no activity also. So here see another examples here, anti-cancer drug, mostly used anti-cancer drug, that is paclitaxel. So these are having 11 chiral centers. You see here. So due to presence of more number of chiral centers, they are more potent and used as an anti-cancer drug. So due to presence of these chiral centers, they are more biologically active. Next we'll go for chiral carbons, another example of thalidomide, but it is now banned due to some side effects. But due to this chiral center, it is also a biologically active drug. So these are some of the examples of drugs which contain chiral carbons. So it is essential for a drug to be biologically active. So these are some of the chiral drugs containing useful for the anesthetic purpose. So you can see the examples. So these are the drugs which are having single isomers or single chiral centers, you can say. So these are some other drugs. So these are categorized in different types. So these are used for anesthetic practice or anesthetic purpose, which are containing chiral drugs. So today we have discussed up to this. So you have any doubt, you can ask me and raise your hands. Tomorrow we'll discuss remaining parts of the stereochemistry like enantiomers, diastereomers, or any other examples also related to this matter. So you can raise your hands if you have any doubts. Shivani, unmute yourself.
Sir, I explain the plane polarized light again.
Okay, 1 minute. So this is your general ordinary light, okay? Okay, this is your plain ordinary light, which are having multiple wavelengths. You can say they are vibrating in different wavelengths. So that's why they are polychromatic light, or they have polychromatic radiations. When they will allow to go through this polarizer, so the polarizer has capacity to convert polychromatic light to monochromatic light. Is okay? So polychromatic light will be converted to monochromatic, means they have single wavelength. Okay. So then when it will pass through a tube containing your chiral carbon or chiral compounds, so they have the capacity, this compound have capacity to rotate the plane of polarized light towards right or towards left. So based on the direction, you can assign as a plus form or minus form or plus D or plus L. So this is the role of plane of polarized light, how it relates to the biological activity. But here you can see without chiral carbon, it is not having any rotation capacity. It cannot rotate the plane of polarized light towards right or left. So it is not having any biological activity. Is it clear to you? Shivani? Shivani, where? You raise your hand again. Shivani is there. Shivani is not showing here. Shivani, again raise your hand. Shivani is there. Not showing here. Okay. Next, Oliva is there. Oliva, ask your question. Shivani, any doubts? Hello.
Yes, sir. Yes, sir, I got it.
Okay. So tomorrow I'll discuss some questions regarding this. You have any doubt, you can clarify again. Okay. Oliva is there. Oliva, you ask your question. Any doubt is there?
Sir, which is more stable in cis and trans?
I have already told you. Okay, we'll discuss again 1 minute. Cis and trans. Due to presence of bulky groups, you can say here. 1 minute. That is slow. Okay. Here you see the examples here. I can clarify here. So here both are bulky groups present on same side and present on very closer towards each other. So they can repel. This more steric repulsion is there on same side. But here you see in transform, they are present away from each other. Means the distance between both the groups are more. So they are unable to, means they cannot repel. This steric repulsion is very less in case of transfer. You can see the diethyl stilbestrol also. You see the distance between both the groups. They are away from each other, very long distance. But here you see in cis form, both are in same side. So the bulky groups definitely cause steric repulsion due to less distance between them. Is it clear? Oliva, it is clear? Hello? Oliva is there? Oliva, it is clear? Hello?
Yes, sir. It's clear.
Okay. Any other doubt you can ask directly, otherwise again you will be unmuted, muted.
No, sir.
Okay, tomorrow we'll discuss some questions regarding this. So any doubt you can clarify also there. Okay.
Okay.
Shivani, any
Thank
doubts there?
you, sir.
Okay, thank you. Shivani, any doubts again?
No, sir, no.
Okay. So now all are attending or not, your batchmates? All are coming
Yes,
here, all batchmates or seniors are there?
All,
Okay.
sir.
Okay. Anybody, any questions you can send on chat box also. I'll make ready. Tomorrow I will give the answers otherwise. If you are unable to ask any question, you can give query on chat box on Zoom. So I will give the answers. Shivani is on hand. Shivani, lower your hand. Now you have no question, na? Okay.
There is not going what.
Okay. Now you are expert in all the digital technology, huh? Okay. Anybody? I forgotten your friends' names. Because somebody somewhere there. 54 today present all. So you for GPAT purpose, you remember some examples of drugs related to stereo part, okay? Maybe cis trans or EZ system or RS system. So specific example of drugs is mainly required for your GPAT purpose, okay? Because organic compounds are not useful now, but drugs related examples is more useful. Hello?
Yes, sir.
Any other doubts?
No, sir.
Who is there in your classmates? Anybody is there? Okay, nobody has no doubt. So tomorrow again we will meet at 10 o'clock. Okay. So anybody has any doubt, we can clarify tomorrow. Okay.
Okay, sir.
Okay. Thank you. Thank you. Okay.
Thank you, sir.
Okay.
Thank you, sir.
Okay.